Um, we'll be presenting the impact of human activity on protected versus unprotected rocky intertidal zones in San Mateo County. I'm Sally Han, and my other presenter is Brandon, and our advisor is Connor Adam. Next slide. First off is our abstract. So our group seeks to investigate the impact of the change in human activity on Northern California rocker intertidal ecosystem. Our research centers around the question, how has human interaction with rocky intertidal zones affected their ecosystem health, as well as species biodiversity and abundance when compared between protected and non-protected marine areas? Due to humans being able to take species from non-protected sites, the ecosystems of the non-protected type have likely been severely affected. Some species that have been removed are moon snails, shore crabs, rock crabs, and even hermit crabs. An example of how species being taken could ruin an environment is sea urchins. Sea urchins are predators to algae and plants, and now that they are being taken by humans, many species of seagrass and other organisms have grown dramatically due to the non-competitive environment. And while this is all happening, overfishing is also largely unregulated, which leads to much more damage in tide pools as well. To start off our research, we first went to Mavericks Beach, which was a non-protected area, and there we took quadrat data using random sampling and transect lines. And then after collecting data from Mavericks Beach through multiple trips, we obtained permits from the Fitzgerald Marine Reserve and began data collection for the protected area. First off, our location was Mavericks Beach. It's an unprotected marine area, so we didn't have to obtain permits to go on there. And then our second area was Fitzgerald Marine Reserve, which is a marine protected area, so we had to obtain permits to visit. And then we'll be now introducing our introduction and background. So our group seeks to quantify the human impact on California Half Moon Bay tide pools in two different conditions, non-protected versus protected areas. The human influence is primarily from trampling of tide pool habitats and harvesting of tide pool organisms. Former is rather straightforward, with all visitors being able to kill, injure, or displace organisms. Latter may occur legally through anyone with a fishing license, provided that is not closed off by local authorities or regulated as an MPA. Since MPAs restricted visitors, there has been an absolute stop of human take and trampling, which resulted in less disturbance of tide pools, which increased biodiversity and species abundance. Our hypothesis is that increased human activity in non-protected areas will reduce the biodiversity average size and abundance of local species when compared to the not protected area. The objective of our group's project is to quantify the effect of human disturbance on tide pools by comparing the counts and sizes of biodiversity between marine protected tide pools and unprotected tide pools. While there's been extensive research conducted on tide pools throughout the years, our project specifically hones in on how human taking of Northern California tide pool species has the larger effect it has on intertidal ecosystems. Next slide. And then now I'll be introducing the methodology. Okay, yeah, I'll pass it off to Brandon now.
Okay, so to further explain our methods from the abstract and stuff, we visited the unprotected tide pool area in Mavericks Beach twice, and the protected area in Fitzgerald Marine Reserve twice to conduct random sampling on species counts, sizes, as well as abundance and biodiversity quantifications in the area. We visited high, mid, and low intertidal zones to collect a variety of data sets, and due to time restraints, we couldn't hand count the quadrats we set out, so we instead used ImageJ software in order to analyze them later. To place our quadrats, we first set out two transect lines serving as our x and y axis. Our x-axis was 50 meters in length and our y-axis was 30 meters. We used a random number generator to find several coordinates along these axes to eliminate human bias. And then we set down 50 by 50 centimeter quadrats at each of these coordinates. We took several photos and started with a picture of the entire quadrat, then zooming in on individual portions. This allowed us to gain a more detailed view of the area, increasing our accuracy. Many of our quadrats were also covered in algae and seagrass, so we moved these to get a better view, and we included a ruler as scale. Possible sources of error with this process was tilt with the cameras, as we needed a camera to be positioned at a certain angle for each image. So it was difficult to confirm whether the camera did or did not tilt for each picture that was taken. Another possible source of error was if the ruler was or was not visible in the picture. For example, if the ruler was face down or only if the edge of it showed, our scale may not be correct. And to remedy these sources of errors, one of our group members would hold the camera still while another moved and cleared up the quadrat. In addition, we checked photos afterwards to make sure the ruler was there. Lastly, circumstances out of our control could have altered the time we spent at each tide pool. If low tide was at close sunset or it rained, we would miss out on chances to get more comprehensive data. After collecting pictures, we analyzed them using ImageJ. ImageJ allowed us to measure the number of species in our quadrats and the species sizes. This gave us a better idea of species abundance in intertidal zones, and we put this data into a master spreadsheet. We also used the program RStudio in order to generate graphs and run statistical tests on our data. Here are some of the animals and plants at these locations, and you can see them in our quadrats. For results, Mavericks had 19 unique species, while Fitzgerald's had 20 different species. So what do these numbers really mean? So we can use the Simpson's biodiversity index, and we found that for Mavericks, it had a value of 0.5365, while for Fitzgerald, it had a value of 0.3382. So this means that Fitzgerald had a higher species diversity than Mavericks, as the further away from zero the Simpson's biodiversity index number is, the lower the diversity. Seagrass coverage for the quadrats were in general that Mavericks had a 42.25% surface area coverage, while Fitzgerald is still being processed. We can visually tell that the location has generally lower surface area coverage from seagrass. Another prevalent animal or organism at these areas were snails. We used a chi-square test with each quadrat acting as a data point. When performing the test, we got a p-value of 0.02. This passes the threshold of 0.05, meaning these results point to the differences in density as being statistically significant. When looking at the abundance of certain key organisms, such as Tegula funebralis, commonly known as the turban snail, Maverick's tide pool had an average density of 36 turban snails per meter squared, while Fitzgerald had 33. The size of the species were also different, as it was an average of 1.774 at Mavericks, whereas Fitzgerald had an average size of 0.949 centimeters. Here are some graphs of our data. And in conclusion, in the upcoming semester, we hope to move on to data analysis and switch from ImageJ to R. R will allow us to get more descriptive data and figures and from the quadrats we've analyzed. Currently, before further data has been analyzed, Maverick shows a higher diversity of species than Fitzgerald, and snail sizes and density show a severe increase in size in Mavericks, suggesting that Maverick's size may have increased due to a decrease in competition, and Fitzgerald generally shows an increase in seagrass coverage per quadrat as compared to Mavericks. We believe that our results may have a significant impact on how local park rangers regulate the harvesting of tidepool species, and the results can provide insight into what is and what is not beneficial to the health of tide pools, and may help rangers revise the list of species that can and cannot be harvested. Thank you, ASDRP and Mr. Adams, for your help. And we have a bibliography with our sources.